Thank you, Chani. So we have a question on the wiki. Very good presentation. I do want to ask why you are going towards genetics. Are there other biomarkers not good enough? For example, can't the plots in the first few slides you showed as background cannot be explained by parameters such as age?
Can you hold on? I can't see the.
Sorry.
Broke up a little bit. So the question was about using other biomarkers. I think this is a great question. So heritability, or specifically neurosense heritability, is what we refer to when we talk about the proportion of variants of a particular phenotype, in this case CAD, being explained by genetics as opposed to environment. So of course, age, which is a combination of genetics and environment, actually it plays a big role. We think that in the United States, because neurosense heritability actually is population specific, that CAD heritability is at 35 to 50%. And so that means that there are genetic contributors that could be identified. And in terms of biomarkers, I think really the work that I'm doing is kind of the first step, which is that genotypes are really affordable to measure, just literally not only millions of SNPs directly, but then also impute millions more. And being able to have access to data from 100,000 individuals, for example, will allow us to identify genes that then, yes, eventually could be used as a biomarker. I don't anticipate that someone who's worried they might have a heart attack would one day walk into a clinic and say, like, I mean, they might, but say, genotype me. I think if someone says, I think I'm having a heart attack, you know, you measure troponin, of course, but there might be other things that you could use in the kind of preliminary phenotypes that before that final stage that this will allow us to detect.
Thanks. I actually have a question, Chani. So in all the diverse data types that you're bringing together, what are your greatest data science harmonization challenges? How do you harmonize this data?
One of the main issues that we're finding is that, you know, of course you want to include as many people as you can in your study sample, not just for power, but because they agreed to be included. And one issue that we've had in the Miller lab is that we're comparing RNA sequencing data with low pass whole genome sequencing, which is basically a genotype, and then also ATAC-seq data. We're looking at chromatin accessibility. And sometimes those samples don't meet QC, and so you have to exclude someone from your final analysis because they had 2 pieces of data, but not all 3 for kind of complete set efforts. That's kind of my biggest shortcoming right now. I wish that we were able to have more access to tissues. One thing I didn't mention is that tissue specificity is really important. Nobody donates their coronary artery tissue while they're using it. And so that's a severe limitation for us, but I think also something that's a strength is that we, you know, kind of held out for this appropriate sample type that we have, as opposed to saying, let's look at EQTLs and, you know, white blood cells from a blood draw and see what we can find. Did that answer your question?
Yeah, thank you. All right, so let me unmute everyone and let's give a round of applause to everyone.